Hello, my name is Dr. Cesar Rodriguez-Saona. I'm the cranberry entomologist at Rutgers University. Here is my email in case you need to contact me. crodriguez@njas.rutgers.edu. Today I will talk about Cranberry IPM or Integrated Pest Management. Cranberry is a crop native to North America and is an important crop in the United States. The United States is the largest producer of cranberries, and cranberries rank fourth among the most important commercial berries and has a value of about $225 million. It is now produced in other countries in North and South America and in Europe, and is consumed mainly due to its known health benefits since it contains high levels of antioxidants. In the United States, New Jersey ranks third or fourth, depending on the year, in cranberry production after Wisconsin, Massachusetts, and Oregon. Cranberries are produced also in Canada, Chile, and Turkey. Most cranberries in the US are produced in the states shown in red, on the West Coast, Oregon, and Washington, and in Wisconsin, Massachusetts, and New Jersey in the East Coast. Cranberries are woody evergreen perennial plants. The plants are low-growing vines, and this is an important fruit crop in New Jersey. As I mentioned, New Jersey ranks third or fourth in cranberry production in the US, depending on the year. This is a unique crop grown on acidic soils and that requires water for harvesting and frost protection, and also relies on pollinators, including commercial honeybees for pollination. The majority of cranberries produced are destined for processing, including products such as juice and craisins. Most cranberry growers belong to Ocean Spray in our state. This is a grower-owned cooperative established in 1930 that plays a major role in cranberry production and marketing. Several issues have caused changes in pest management practices in recent years. These include the fact that cranberries are a specialty crop grown in limited acreages in the United States, the passing of the EPA Food Quality Protection Act that restricts use of certain broad-spectrum insecticides. This is critical since growers have relied on organophosphates and carbamates to control certain insect pests. Thus, there is the need to develop alternatives to chemical control. These changes have impacted pest management practices in cranberries, including the reassessment of tolerances for broad-spectrum insecticides, measures to reduce worker exposure, limits on the number of applications, maximum residue levels, pre-harvest and re-entry intervals, and the development of new management tools. Currently, most cranberry production in New Jersey incorporates aspects of integrated pest management, reflecting the progressive approach of our growers. However, there remains a need for chemical controls and for developing alternatives to broad-spectrum insecticides. Our IPM program uses a variety of monitoring methods, including sweep netting for early season Lepidopteran pests, pheromone-baited traps for tracking Lepidopteran populations, and visual inspections to assess damage to fruit and foliage. Only a few established economic thresholds exist, and they are based on counts from SweepNet samples. We recommend taking sets of 25 sweeps. Our thresholds are 1.5 black-headed fireworms or Sparganothis fruitworms per sweep set, and 4.5 larvae of armyworms, blossomworms, or spongy moth larvae per sweep set. In a recent publication, we searched the literature for all the papers on cranberry pest management for the past 30 years. We used peer and editor-reviewed publications obtained from Web of Science and arthropod management tests. We identified about 900 publications, but removed half because of duplication. We ended up with 677 publications and used 139 that met our criteria of insect pest management in cranberries. From these publications, we extracted the location of the study, the pest that was studied, and the IPM tactic used. We found that most studies are from North America, including the United States and Canada, and within the United States, most studies originated from Wisconsin, Massachusetts, and New Jersey. These are the major cranberry producing states. So this data correlates with the top cranberry producers. The most studied insects include the black-headed fireworm, Sparganothis fruitworm, and cranberry fruitworm. All of them are native to North America and are direct pests that can damage the fruit and reduce yields. Blind-nosed leafhopper, a vector of a disease, has also received great attention. So these graphs summarize all the studies across different IPM tactics. Clearly, most research have been done on chemical and natural and biological control, and fewer on behavioral and cultural control, and also in host plant resistance. Cranberries in New Jersey are susceptible to at least 10 insect pests throughout the season. This chart shows when growers should monitor for this pest in gray, and in black when control treatments should be applied if needed. These pests attack different parts of the plant. There are pests that attack the buds, the leaves, the flowers, or fruit. There are also insects that attack the stem and others that attack the roots. 4 major pests that attack the buds, the leaves, flowers, and fruit in New Jersey are spotted fireworm, cranberry blossomworm, Sparganothis fruitworm, and black-headed fireworm. The spotted fireworm is one of the most important pests in New Jersey cranberry farms. This insect overwinters as second instar larva. It has 2 generations a year, and the females lay their eggs in masses on several oviposition hosts. The population can be regulated by natural enemies such as egg parasitoids, and fruit can be damaged by second generation larvae. Eggs are laid in masses and found mainly in weedy areas. The eggs are laid on maple, briar, leatherleaf, loosestrife, and other weeds. These egg masses can experience high parasitism rates by egg parasitoids in the genus Trichogramma. Cranberry blossomworm has a single generation per year. The eggs are laid from September through October and overwinter, hatching the following May. The mature larvae can reach large sizes. By late May, larvae turn nocturnal and become very voracious. This insect has a long pre-pupal dormant period, and pupation takes place in late July through early August. The moths emerge in September and are active until November, and the females can lay their eggs singly and can lay between 100 to 200 eggs. Sparganotis fruitworm is an important pest in many cranberry-producing states, including New Jersey, Wisconsin, and Massachusetts. This insect overwinters as early instar larvae and has 2 generations per season. The larvae from the second generation can feed on cranberry fruit. The eggs are laid on cranberry leaves, and the larvae also feed on several weed species. There is evidence of populations that are resistant to organophosphate insecticides. Black-headed fireworm is another important pest in many cranberry-producing states. This insect overwinters as eggs and produces 2 generations per season. The larvae from the second generation can feed on cranberry fruit. The eggs from the first generation hatch during bloom, and several egg parasitoids are recorded. Black-nosed leafhopper is a key pest of cranberries in New Jersey. This native insect feeds on several host plants, mainly in the Ericaceae, the family Ericaceae, and is considered a pest because it vectors a phytoplasma, a type of bacteria that causes false blossom disease. This disease is characterized by malformed flowers, causing the flowers to stand erect, and by petals rather than normal. Because the phytoplasma infected plants are alive but sterile, and their only purpose is to vector the disease, they have been referred to as zombie plants. False blossom disease has a bad reputation in New Jersey. It has been blamed as the cause of a decline in cranberry production since the 1950s. This insect has a single generation a year. It goes through eggs, 5 nymphal instars, and the adult stage. The insect overwinters as eggs, and the eggs hatch in May. Adults are most abundant in July, and the eggs are laid in July through August. The one major pest that feeds on the stems is the cranberry girdler. This insect has a single generation per season. The larvae girdle the stems, and severe infestations can cause patches of dead vines. It overwinters as a full-grown larvae within a cocoon and pupates in May, and the moths emerge in June. The eggs are laid in sand and litter layers. Regular sanding suppresses or retards development of girdler. 2 insect pests that feed on cranberry roots are cranberry rootworm and white grubs. The cranberry rootworm has a single generation a year. The larvae feed on tender roots, and the adults feed on foliage. It overwinters as fully mature larvae. Both the larvae and pupae are susceptible to entomopathogenic nematodes. There are multiple species of white grubs, primarily on the genus Phylophaga, that feed on cranberry roots. These insects have 2 to 3 year life cycles. The adults emerge in May through July. The females deposit their eggs in the root zone and feeding by larvae can cause the death of vines, and this damage can be seen as dead patches. Chemical control is recommended after bloom to control the larvae in the soil. This information that I am providing you is available to our growers through our extension program at Rutgers University. This program is built on regular interactions with growers. For example, I serve as a liaison for the American Cranberry Growers Association and help organize their meetings, including annual field days. To provide information on pest management through I also provide information on pest management through seasonal blogs, fact sheets, annual recommendations, meeting proceedings, and through my website. We have been involved in several research projects to improve our monitoring tools, assess the efficacy of beneficial organisms such as entomopathogenic nematodes, and to investigate the effects of disease infection on cranberry resistance to insect herbivores. For example, we have tested the attraction of leafhoppers to color sticky traps. This is important because there is no available technique to attract adult blind-nosed leafhoppers. Current monitoring relies on the use of sweet nets to monitor for the nymphs. However, this can be labor-intensive. Also, the nymphs are small and difficult to detect. Additionally, yellow sticky traps are used to monitor sharp-nosed leafhoppers in blueberries. So we tested 6 colors in 5 cranberry beds, and we counted the number of leafhoppers weekly from June through August. We found that sharp-nosed leafhoppers were highly attracted to yellow. In contrast, blind-nosed leafhoppers did not show a strong preference for any color. Among the colors tested, red was the most attractive to blind-nosed leafhoppers. White grubs are also difficult to control because only a single insecticide is registered for their management. This limitation raises concerns about the potential for the development of resistant populations. For this reason, we first conducted a study to improve their monitoring. We tested traps baited with the pheromone L-valine methyl ester, and we were able to capture Phylophaga georgiana, the principal Phylophaga species in New Jersey. This species was captured in the pheromone-baited traps from June through early September. We also tested entomopathogenic nematodes as alternatives to chemical control. Cranberries are a suitable crop for the use of entomopathogenic nematodes because of the high soil moisture levels and relative humidity, the protection of the root zone from direct sunlight, and temperatures that rarely reach levels harmful to the nematodes. We tested 3 entomopathogenic nematodes using potted cranberry plants. We tested their efficacy in controlling second and third instar larvae. We found that one of these species, Sternenema scarabi, was the most effective against both instars than the other 2 nematode species. We then tested lower rates from 1 to 0.125 billion per acre and found that the nematode was effective even at low rates compared to the untreated control. In my lab, we're also studying the interactions between cranberries and insect herbivores. This includes insect vectors of diseases and non-vector insects. But infection by a pathogen can change the phenotype of the plants. And these effects of pathogens on the plants can affect their interactions with herbivores. Thus, we're exploring the chemical ecology. We're exploiting chemical ecology to better understand the interactions between the cranberries, the phytoplasma that causes false blossom disease, and the vector, the blind-nosed leafhopper, and non-vector herbivores. One pathogen that is present in cranberries are, as I mentioned, phytoplasmas. These are a type of bacteria that lack a cell wall. They infect the phloem cells in plants and are transmitted by phloem feeders like leafhoppers. We already discussed blind-nosed leafhoppers that vector a phytoplasma that causes false blossom disease. This disease is characterized by malformed flowers, causing the flowers to stand erect, and by petals rather than normal. I also mentioned that because the phytoplasma infected plants are alive but sterile, and their only purpose is to vector the disease, they have been referred to as zombie plants. So we have been conducting no-choice experiments with early instar nymphs placed in plastic cages to look at the effect of uninfected and phytoplasma-infected plants on the mortality and weights of the leafhoppers. We found no effects of phytoplasma infection on nymphal weights. However, the weights of adults were higher in infected plants compared to non-infected plants. We found no effects on leafhopper survival. We then conducted experiments to look at attraction of nymphs and adults to infected and uninfected plants in an olfactometer, and also looked at oviposition preference in cage studies. In addition, we collected volatiles from these plants to look at the differences in volatile plants. So we found that nymphs are attracted to odors from uninfected plants, but not to odors from infected plants. However, when given a choice between infected and uninfected odors, there was no preference. Similarly, adults were more attracted to odors from uninfected plants, but not to those of infected plants, and show no preference for odors of the two when offered together. In single-choice cage studies, females prefer to oviposit on uninfected plants than on infected plants. And this was the same when given a choice. So in addition, as I mentioned, we also collected volatile emissions using head-space volatile collections and analyzing the samples using gas chromatography mass spectrometry. We found that higher volatile emissions from plants that were healthy compared to the phytoplasma infected plants. In conclusion, we found that in support of the host manipulation hypothesis, there was no effect of the infection on survival or nymphal mass. However, the adults were bigger on the infected plants. The nymphs and the adults were attracted to healthy plants, and there were less eggs that were laid on the infected plants. And the infected plants produce less volatiles. We are also doing experiments to understand the interactions between cranberries, the phytoplasma that causes false blossom, and non-vector herbivores, such as caterpillars. So in these studies we conducted, we looked at the effect of infection on different species of caterpillars. We looked at spotted fireworm and Sparganothis fruitworm, two native insect pests of cranberries, and also we looked at the invasive spongy moth. We placed these caterpillars in bags with infected plants or uninfected plants. And then we assessed their mortality and their weights. We found that all the species of caterpillars were bigger on infected plants than non-infected plants. And also, all of these caterpillar species ate more of the infected plants than the non-infected plants. We measure various macro and micronutrients in the leaves and found that many of them were higher in infected plants. In particular, nitrogen levels were increased in infected plants, which could explain the improved caterpillar performance. We also measured levels of proanthocyanins that are important defensive compounds found in cranberries, and found that the levels of proanthocyanins were lower in the infected plants. In conclusion, we are finding that infection affects non-vector herbivores. We found that larvae are bigger on infected plants. They consume more leaves on the infected plants, and also that infected plants have more nutrients and also are less defended. So in summary, these studies show that the phytoplasma likely manipulates the levels of nutrients and defenses in cranberries for its own benefit, and that it has consequences with the plant's interactions with vector and non-vector herbivores. In fact, what we are seeing is that infection reduces the levels of defenses in the plants and increases the levels of nutrients. And this results in increased weights of adults of the vectors of the disease, the blind-nose leafhoppers, and also increases in the larval masses of 3 different species of Lepidoptera. I invite you to visit my website to learn more about my program. This site is linked, the site that I'm showing you is linked through the Rutgers B. Marici Blueberry and Cranberry Research and Extension Center. I also have a personal website, and you are welcome to contact me if you have any questions. Thank you.